Okay, we're going to start off pretty quickly today so that we don't get too behind on our final unit. Only got a very little bit of class left. So a reminder, there will be a quiz this weekend. It'll go up this afternoon, so stay tuned for that. I'll try to send out an email once that is up. But that said, let's get rolling and continue with the GI system. Okay. So when we left off, we were talking about the stomach. All right, so we talked a little bit about the physical structure of the stomach, which you saw in lab. It has these folds inside, allowing it to expand. These folds are called rugae. It's going to contract to mix together food and this product of the stomach that we call gastric juice, which is made up of mucus and a bunch of different types of enzymes, which is what we're going into right now. So today we're going to be focusing on enzymes that help us digest and break down our food. Okay, so the gastric juice that's mixing together with food to create chyme is coming from these cells in these structures called gastric pits that can be found in the lining of the stomach. So the lining of the stomach would be like here, right? So here we see that same border, but diving into the tissue, we have these gastric pits. So this is a gastric pit, so it would be located kind of deep here, but they're small, which is why they're not in the bigger picture. Okay. So the purpose of the gastric pits is to produce the different components of our gastric juices. And basically what they kind of are glands. So these glands are located kind of in the bottom of the gastric pits. Okay. Someone's joining us, sorry. Okay. So the gastric glands there at the bottom are going to be secreting different products that help us digest food. So the stomach is going to be the beginning of our digestion of proteins. It's going to help us continue digesting with its acidity. It's going to help us continue digesting our carbohydrates. And it's basically going to be producing a very acidic environment that kind of helps us break down food. Okay, so there are a couple different products that we want to know about. So, and they're going to link up to different types of cells in the gastric glands and the gastric pits. So we can see we've zoomed in here on one of our gastric glands, and we have it broken out on the right here. Okay, so we can see our chief cells, which are here in beige, and we can see parietal cells interspersed a little more distantly in purple. Okay, so our chief cells are going to be producing pepsinogen. So pepsinogen, from this prefix pep, we may kind of recognize. So pepsinogen is going to be the start of our pathway for digesting proteins. This inogen may be vaguely familiar to us from when we talked about clotting, right? We have seen this ending before, and we know that this is usually telling us that this is a molecule which is going to need to undergo a conversion step before it becomes an active enzyme, which is the case for pepsinogen. So pepsinogen, we'll see, is going to turn into pepsin, which is the enzyme that pretty immediately digests the proteins. So that's coming from our chief cells. But our other cells here in the gastric pits and in the gastric glands are doing things too. So those parietal cells are producing two different products. So the parietal cells are going to be producing hydrogen ions. So the hydrogen ions are going to maintain the stomach as a very acidic environment. So we're basically going to have hydrochloric acid in the stomach. And that hydrogen is coming from the parietal cells. The other thing that the parietal cells are secreting is something called intrinsic factor. So intrinsic factor is there in order to help us absorb vitamins, specifically vitamin B12. So in order to absorb vitamins, we need the help of this molecule as well. So intrinsic factor and hydrogen ions are coming from the parietal cells. The hydrogen keeps the stomach acidic. The intrinsic factor helps with B12 absorption. The gastric glands and gastric pits are also going to be helping us secrete a hormone called gastrin. So you don't see it in this image. It's secreted from something called G cells. So gastrin is a hormone, so it's going into the bloodstream. And gastrin actually goes all the way up to your brain. Gastrin is a hormone that's kind of related to hunger and satiety. So telling how much food you have in the stomach when you want to eat. There are a number of different hormones that have this kind of interplay that regulate your hunger cues, and gastrin is one of them. So that's coming directly from the stomach as well. Now finally, because the stomach is so acidic, we also want to protect our tissue from that acidic environment. So we have mucus lining the stomach as well. Mucus is coming from cells a little farther up in those gastric pits. So it's coming from these neck cells, so up kind of at the top near our mucosal layer. So that's what's lining the stomach. Okay. So this creates a really acidic environment full of different precursors to enzymes, full of products that are going to help us with digestion and absorption. Now, the acidic environment of the stomach is one of the things that actually lets us activate pepsinogen. So that acidity coming from those hydrogen ions, which are a product of our parietal cells, is actually going to activate the pepsinogen from the chief cells. So we really need both in order to end up with our active enzyme pepsin, which is going to start to break down our proteins. Now, the acidic environment of the stomach is also participating kind of directly in the digestion of proteins because just the fact that it's so acidic starts to denature proteins, by which we mean it kind of starts to unfold them from their big tertiary and quaternary folded structures. It starts to denature that structure, unfolding it and starting to break them down into their sort of linear polypeptide chain shape. The acidic environment of the stomach is also important for immune function, kind of. So the acidity of the stomach, it's at a pH of about 2. That's very acidic. And it actually kills a lot of bacteria. So not that you're never going to get sick from eating spoiled food with bacteria in it, but your stomach is doing a fair bit to protect you from that just through its own acidity. And so it also has this gastric mucosal barrier protecting us from that acidity, as we mentioned. So we have mucus secreted from the neck cells. That mucus is actually mixed with bicarbonate. And we know from our discussion of acid-base balance, which we've been discussing earlier in the week, that bicarbonate kind of soaks up hydrogen ions, right? Its base, it kind of neutralizes them. So the bicarbonate in the mucus stays kind of stuck in the mucus, but as the hydrogen ions come into contact with it, kind of neutralizes it directly on the tissue, preventing you from digesting your own stomach, basically. There is another type of cell that's also involved in the production of this mucus and bicarbonate. They're called goblet cells, but they're also not on this image. Okay. That moves us into the gastrointestinal tract. So the gastrointestinal tract, and specifically the small intestine, are going to be adapted for absorption. So they're specialized for absorbing nutrients from the lumen into the bloodstream. So thus far, our food has been traveling down the pipe, right? Traveling into your stomach. We've done a little bit of digestion. We had our salivary amylase. So we had an enzyme that breaks down carbohydrates in the mouth. And we had this pepsinogen turning into pepsin in this very acidic environment of the stomach, kind of starting to break down our proteins. But we haven't actually absorbed these nutrients, right? We've just started to break them down. They haven't started moving into our cells. That's what's going to be happening in the small intestine. Most of the absorption actually happens in the first 20% of the intestine length. And the small intestine as a whole is 8 to 10 feet long, right? So maybe the first 2 feet or so are going to be doing most of the absorption. But we have more intestine allowing for further absorption if necessary. The structure of the small intestine as a whole is anatomically arranged to provide a really large surface for absorption. And we mean that on both a kind of macro level with this long length. When you saw in the body, right, you saw small intestine was really coiled, lots of it crammed in there into a really small space. If you kind of took the small intestine through your hands and moved it through, you kept going and going and going. So that provides a large surface area. The other thing that we're going to see providing a large surface area in the small intestine is the border of the cell. When we look at them, they're going to have a lot of microvilli on the apical membrane, creating a brush border. Again, providing a really large surface area for absorption. So as you saw in lab, the location of the small intestine, right, after the stomach, but before the colon, right, and it's very highly coiled. Now I can't follow exactly where the coils are here, right, but it's all swirled in there, eventually leading to our cecum and then leading to our colon, which is larger in diameter but shorter in length. Okay. So the small intestine has 3 divisions: the duodenum, the jejunum, and the ileum. So hopefully we remember that from lab. Okay. So now we're going to look a little more in detail at the microscopic structures that we would see in our small intestine. So here we're looking at a cell, right? Over here. So we're looking at some small intestine cells. And here is the shape of the cells themselves. All right, so zoomed in once and then zoomed in twice. So on the apical membrane, we can see all these projections. These are our microvilli. Right, so this is increasing the surface area of the epithelium of the small intestine. And in between these epithelial cells, right, we also have these projections of the cells themselves, right? So what I mean is, right, the cells themselves have microvilli, and then the cells are structured in these kind of projections. Okay, so these projections contain blood vessels and a lymph structure called a lacteal. So here we see, right, an arteriole coming in. We see a capillary bed leading to a venule, leading to a vein. So that's our blood flow. And we can see in the center, we have a branch of our lymphatic system, right? So we have this lacteal leading to a lymph duct. Now, we haven't talked a lot about lymph ducts before, but that's going to be important as we start to talk about digestion of fats and absorption of fats. So that's going to be the role of those lymph ducts. Stay tuned. The other thing we want to know about in the walls of the intestine are something called the Crypts of Lieberkun. Okay, so these are basically going to contain epithelial cells that are secreting bicarbonate. Right, so the fluid, the chyme, coming from the stomach, right? The stomach had a pH of 2, right? So we have a very acidic environment in the stomach, but the small intestine, its contents are not acidic. So we have bicarbonate-rich fluid being secreted in the proximal parts of the small intestine. So the parts right next to the stomach, right? So like the duodenum, are secreting bicarbonate in order to neutralize that stomach acid. So that's going to continue down. By the time we get to the more distal parts of the small intestine, so like once we're in the ileum or something, all right, we don't need that bicarbonate anymore. We've neutralized the stomach acid, so it will be reabsorbed in the distal parts of the small intestine. So we're going to talk a little bit about the pieces of the small intestine specifically, mainly the duodenum. So the duodenum is coming right after the stomach, right? So here we see the stomach. We have this region called the pylorus, and we had the pyloric sphincter, which you looked at in lab. And then we have the duodenum or duodenum. Okay. Now this is an important location because this is where we are going to be receiving pancreatic juice and bile. So pancreatic juice and bile are coming into this first part of the small intestine, the duodenum. The pancreatic juice is going to contain a bunch of digestive enzymes, right? So lots of different types. There are actually digestive enzymes coming from the pancreas that break down all 4 of our different macromolecule types. The pancreatic juice also contains bicarbonate, neutralizing the chyme, right? So bicarbonate coming from those Crypts of Lieberkun is going to be coming directly into the duodenum to help us neutralize the acidity of that stomach acid, leading acidity, giving us acidity in the chyme up until this point. Okay. The other thing we have coming in at the duodenum is bile. So bile is going to be important for breaking up fats in order to allow you to absorb fats, which fats have kind of their own thing going on as we talk about how different macromolecules get broken down, because we remember that fats or lipids are nonpolar, right? So they're going to interact very differently with the contents of the intestine than our carbohydrates and proteins, which do have polar parts and interact differently with water and with the cell membranes here. So bile is coming from the liver and sometimes the gallbladder, which is kind of a storage organ. Right? It's going to be entering the duodenum, mixing with the chyme coming from the stomach and the pancreatic juice coming from the pancreas. The bile is going to contain a lot of these things called bile salts. And as we talk about lipid digestion specifically, we'll talk about what those bile salts are doing in greater detail. A little bit more about the liver. You've seen it before in lab, but something we want to be aware of as we're thinking about the liver and its role as relates to the GI system is that all the nutrients that we absorb in the small intestine and all our blood flowing through the colon, right, is going to have to pass through the liver. So the venous blood basically coming from all our structures, all our digestive structures in the abdomen, they're not coming into the vena cava directly. They're part of something called a portal system, right? So we have a hepatic portal system. So all absorbed nutrients have to travel into our hepatic portal vein and into the liver, where the liver functions to detoxify substances. It functions to do a little additional processing for some nutrients before it then allows that blood to continue into the systemic circulation and go into the vena cava. So this is kind of a whole separate blood flow system from the rest of this systemic circulation. It has like a kind of control point in the liver. So there is specialized vasculature for this. Okay. So we might start here. Let's say we're starting at the small intestine, right? So we're doing some absorption here at the small intestine. We would first travel into the mesenteric veins, right? So we looked at those in lab, right? So let's say we're going into the superior mesenteric vein here. We would then travel into the portal vein, right, through the liver, where we do detoxification, we do additional processing into the hepatic vein, coming out on the other side, going back to the systemic circulation. Okay. So the liver, therefore, has important arterial flow as well, supplying fresh blood to supply oxygen. Right? So we saw that in lab as well. We have a hepatic artery here coming to the liver. That takes us to the large intestine. So we're not going to go into detail here about cell types really, because the large intestine is really focused on concentrating wastes. So in the large intestine, which consists of the cecum, the colon, and the rectum, right? So cecum, the colon with its four parts, ascending, transverse, descending, and sigmoid, and then the rectum leading to the anus. Okay. It's going to be concentrating waste into feces largely by absorbing water. So we're going to absorb almost all the water that is passing through the colon. And it's also focused on storing feces until defecation. I think you have about usually like 7 pounds potentially of feces stored here in the colon. Obviously, depends on your diet, depends on your body size, depends on your bowel movements. So that number can vary really wildly depending on what's going on in your system, but it can store a lot of mass. The colon empties into the rectum, and then feces gets excreted through the anus. So that's our external sphincter, basically. And the anus is actually made up of 2 sphincters. So just like we saw with the urethra, we had an internal and an external sphincter. Same thing with the anus. We have an internal anal sphincter. It's made of smooth muscle, so it's largely involuntary. We have an external anal sphincter, which allows us to exert more voluntary control over defecation. Both sphincters need to be relaxed in order for defecation to happen, right? You need to open both of these rings in order for anything to get out. Okay. So that leads us to our accessory glands, which are the newer kind of material, right? So the basic parts of the GI system should have been relatively familiar from lab. We've seen the accessory glands in lab, but what we want to focus on in lecture here is a lot more about the products that they're secreting. So the accessory glands are going to be producing different types of enzymes and juices that allow us to digest our food. So to break down our food into molecules that are small enough for our cells to be able to absorb the nutrients. So we have basically 4 types of accessory glands. We have the salivary glands, which we've seen in the head, right? So the mandibular, sublingual, parotid glands, leading saliva to be secreted into the mouth, which is largely made up of salivary amylase, helping us break down carbohydrates. We're then going to see that the pancreas, which sits kind of directly behind the stomach, is going to be secreting pancreatic juice with a lot of different types of enzymes, leading us to be able to digest all of our different types of macromolecules. So secreting that juice and funneling it into the duodenum. Both the salivary glands and the pancreas have similar structures of epithelial cells that allow them to secrete their different types of juices and enzymes. So if you were to slice open a salivary gland or the pancreas, right, and look at it on a slide, you would see that it is structured into these kind of round structures called acini. So these are clusters of secretory epithelial cells, right? So we can see these branching structures with this epithelial lining. So those epithelial cells in the acini are going to be doing primary secretion. So this is just basically directly secreting our initial enzymes into these ducts. They're going to then travel through ducts where they get modified by transport of various ions, transport of water, and then they're going to be funneling into some sort of a main duct. The name of the main duct differs depending on whether you're talking about a salivary gland or the pancreas, but then they are going to eventually get funneled into, secreted into the lumen of the digestive tract, either at the mouth or in the duodenum. Yeah. And finally, the liver has its own kind of structures for secretion, so you wouldn't expect to see acini in the liver, but the liver is also secreting an important product, secreting bile. We're going to go through each of these separately. Don't have a picture of the salivary glands here, but we can think about the product of the salivary glands, which is saliva. It's a spit, saliva. Saliva is rich in bicarbonate ions, so it's a little bit alkaline. Contains mucus to lubricate food, to allow you to swallow it without scratching up your esophagus. And it has different enzymes present. So it has salivary amylase. So salivary amylase is helping you break down carbohydrates. It has something called lysozyme, which has a function of kind of breaking down bacteria. So this is again kind of trying to protect you from any pathogens that might be on your food that you might be accidentally eating. The pancreas has some more specialized structures we want to think about. So the pancreas has both exocrine and endocrine portions, which is to say exocrine, we're secreting some enzymes kind of directly into these ducts in the pancreas, leading into the duodenum, in order to allow us to digest macromolecules. In terms of endocrine function, it means we are going to be sending some hormones from the pancreas into the bloodstream. So the exocrine portion of the pancreas is the part that produces pancreatic juice. So the pancreatic juice, like our saliva, is rich in bicarbonate. So this is largely to help us neutralize that stomach acid that's coming through into the duodenum. Relative to its weight, the pancreas actually secretes more protein than any other tissue of the body, and that's because it's secreting a lot of enzymes. So we have 4 kind of basic types of enzymes that the pancreas is secreting. So there's pancreatic amylase, which is kind of just like the salivary amylase. It's going to be breaking down carbohydrates. We have various types of lipases. So lipases are going to break down lipids. Then we have proteases. Prot comes from protein, ease, it's an enzyme. So these proteases are going to help break down enzymes. And we have nucleases, helping us break down nucleic acids. So we're able to break down all our different types of macromolecules. Okay. So next we have the liver. So that's kind of our third accessory gland, if we think of all the salivary glands as kind of one group together. So the liver has lots of jobs. We've talked just now about how it is filtering all our nutrients, detoxifying, but its gland function is to secrete bile. So as it's secreting bile to help us break down fats, and the bile is rich in bile salts, is also going to be processing some of the nutrients that are coming through that venous blood flow going into the portal system, coming from all the things that are getting absorbed in the small intestine. So way back in A&P1, when we talked about metabolism, we talked about the fact that glucose gets stored as glycogen. And we talked about that when we talked about the absorptive and the post-absorptive state, right, way back in A&P1. So when you eat food, the glucose, the sugars in that food will get stored as glycogen, which is basically glucose molecules linked together, which can then be used later on when you're not directly eating food. Amino acids can also be converted into fatty acids in the liver, right? So these are some of kind of the metabolic functions of the liver. The liver is also creating triglycerides and cholesterol. So it's synthesizing these molecules from other things that are flowing through, nutrients that are coming into the liver. It's also creating lipoproteins. So the liver makes all sorts of stuff, all sorts of weird molecules being made in the liver. Liver has a ton of functions. So when we talked about the cardiovascular system and we talked about blood, we talked about the life cycle of red blood cells. One of our organs that was involved in that life cycle of red blood cells was the liver. So this is just a reminder that the liver is catabolizing or breaking down hemoglobin, creating bilirubin as a byproduct, which is going to get excreted. It's going to mix in with the feces. It's one of the things that give feces their brown color. And as it's breaking down that hemoglobin, right, the reason it's generating bilirubin and breaking down hemoglobin is we want to preserve the iron in the hemoglobin for later use. So that's also going on in the liver. The liver is also synthesizing plasma proteins. So albumin gets synthesized by the liver, and it's secreting and modifying hormones. So basically, I'm not going to say any function you can think of is done by the liver, but the liver really doesn't have one specific function. The liver is really a big multitasker. It's doing a lot of different things. But as we're thinking about the GI system and digestion, a lot of the role of the liver that we're thinking about has to do with its production of bile. So we want to think about the biliary system as we think about the liver. So this is just a kind of quick reminder of what you should have covered in lab before. But we have the liver, right? The liver is where we're actually synthesizing bile, which is full of bile salts that are going to help us break down fat. The bile goes into our hepatic ducts. So we saw that there was a right and a left hepatic duct leading into our common hepatic duct. We also have the gallbladder sitting here. And so the gallbladder, right under the liver, sometimes we're going to funnel bile into there, or we're going to store it for later use. When we want to use our bile salts to help us digest fats, the bile is going to move through the common bile duct. It's going to go into a common structure with the pancreas. It's called the ampulla of Vater, right? So that's this kind of joining point of the pancreatic duct and the common bile duct. They go through a sphincter. So now we have mixed bile with pancreatic juice. They go through this sphincter called the sphincter of Oddi into the duodenum. Okay. So this is going to let us digest fats because of the function of the bile salts and the lipases coming from the pancreatic juice. We have various proteases coming from the pancreas, various nucleases coming from the pancreatic juice, and pancreatic amylase coming from the pancreas. So we're going to expect to see a lot of digestion happening here in the duodenum. Okay. So sorry, I'm pausing because I'm thinking we're going to skip this bit about the liver anatomy because I'm a little concerned for time. So you can write on your slides that we're going to skip exactly how we are creating these bile salts. So don't worry about learning this kind of microscopic anatomy inside the liver here. Okay, so here is just a slide from your textbook. This is an overview to help you study exactly what is happening functionally in each of these organs. So next we're going to move into absorption, and I think these were ordered wrong potentially in your PowerPoint packets, so I want to give you a second to navigate to absorption from the GI system. So we're going to move to the packet that talks about how we're breaking down different carbohydrates, different proteins, the enzymes that are involved that we've just briefly touched on with the glands. We're going to look in detail now at their function as we think about absorption. So I'll give you a second. Okay, hopefully we have that up. Okay, so in this packet, what we're focusing on are the mechanisms by which we're able to digest and then absorb carbohydrates, proteins, and lipids. We're not really going to think about nucleic acids. They're important for your body, but digestion of nucleic acids is not really a big topic. So carbs, proteins, lipids. We're going to kind of relate these when we get to lipids to the different chemical nature of fats, so to their non-polar nature, which is going to lead to pretty different mechanisms of getting them into the body. So we're going to talk about absorbing these three types of macromolecules, and we're going to talk about absorbing vitamins, minerals, and water. So let's start with our carbohydrates. So remember that the structure of a carbohydrate, so we basically have a bunch of monosaccharides. We usually think about glucose, but there are others. So monosaccharides linked together into chains, right? So these chains might be branched like they are in starch, right? So this is a picture of starch, right? Or they might be unbranched, but we have a bunch of glucose monomers kind of linked together. Your body can actually only absorb monosaccharides, so it is only going to be absorbing like individual rings. So when you eat something like a starchy food, right? Say you're eating a potato, you have to break down this big molecule starch into monosaccharides. Now, even disaccharides, even though they look pretty small, they also have to be broken up into monosaccharides. So we're going to go through a variety of steps to break down our large carbohydrates into monosaccharides. Okay. It starts in the mouth. So salivary amylase is an enzyme produced by the salivary glands, so produced in those acini, that allows us to begin to break down starch. Okay, so it would start to break down starch, right? So we're going to break it into pieces. Okay. Pancreatic amylase continues this. So pancreatic amylase is in the pancreatic juice, going into the ampulla of Vater, going into the sphincter of Oddi, going into the duodenum. So right after the stomach, we're picking up this further breakdown of carbohydrates. Okay. So the end products of the digestion of starch with these two enzymes are either little disaccharides, right? Disaccharides, or these structures called limit dextrins. So limit dextrins are produced when we're breaking down one of these kind of branch points. So limit dextrin is a small-ish carbohydrate that includes a branch. So the enzymes have trouble breaking down these kind of corners into disaccharides. So that's when we would produce a limit dextrin. these would then get further broken down into monosaccharides by other enzymes, basically. So we would need to further break these down in order to actually absorb them into the body. So we need to further break them down into monosaccharides for absorption. But we're not going to focus on that for our purposes right now. We're just going to think about the fact that the salivary and pancreatic amylases are making our starch, are making our big carbohydrates much smaller. Okay. Now we're going to turn to proteins. So proteins are going to be broken down by proteases. So a protease is an enzyme that breaks apart protein chains. And we break our proteases into kind of, let's say 3 groups. Whether this counts as 2 or 3 is kind of debatable. But we have our endopeptidases, which I think it may help if I kind of draw as a little protein. So I'm again, think of each of these circles as an amino acid. Right, so we have our amino acids linked together. Then we're going to have the action of our endopeptidases and exopeptidases. So the endopeptidases and exopeptidases are both going to break down parts of this chain, but they break the chain in different locations. So the endopeptidases act on interior peptide bonds, so they might break this like here in the middle. The exopeptidases are going to come in from the ends. So when you have only like one amino acid at the end, that's where you're going to have an exopeptidase come in. So those are our endopeptidases and our exopeptidases, and we'll talk about some specific ones. Then we have Zymogens. So Zymogens, the reason I kind of waved my hands about whether this is 2 or 3. So Zymogens are actually an inactive form of proteases. So Zymogens are going to be stored in granules that have to be secreted by exocytosis and then are going to have to be activated in order to actually act as a protease. So a Zymogen is an inactive protease. They're going to be activated by something called proteolysis. So this is kind of like what was happening with pepsinogen and pepsin in our gastric juice and our stomach acids. So Zymogens are going to have to be lysed a little bit. They're going to have to be broken down a little bit in order to reach their active form. We are going to look at some specific ones. Okay. So the protein digestion is starting in the stomach. Okay. So here we have our protein. And so the first thing that's happening is we're going to have pepsin acting on our protein. So pepsin is secreted by our cells in its inactive form, right? So here we have our chief cell, right, secreting pepsinogen. So pepsinogen is getting secreted into the stomach, where it's going to be activated by the acidic environment of the stomach. So it's going to be activated by the hydrogen ions, essentially coming from the parietal cells. So they're going to act on pepsinogen. Okay. So this creates our active pepsin. Okay. So our active pepsin is then going to start to break down our protein into smaller peptide fragments. So that is our active form. This means that we have basically small protein chunks coming in to our small intestine. Oh, I should have drawn them up in the lumen. Okay, so they're coming along here, our protein chunks. Okay. And let's say we're in the duodenum, because we are, because we can see here our pancreatic duct coming in. Right, so the pancreatic duct coming into the duodenum. Okay. So coming in through that pancreatic duct are all of our pancreatic proteases. So the different enzymes that our pancreas is secreting that allow us to digest proteins. So we have three types, or three proteases we're interested in. We have trypsin, we have chymotrypsin, and we have carboxypeptidase. So these are the three pancreatic proteases. Trypsin and chymotrypsin are endopeptidases, and carboxypeptidase is an exopeptidase. So we can remember that by the fact that it has an X in it. That's not why it has an X in it, but it helps remember it. So endopeptidases and an exopeptidase. Okay. So we also have here in the lumen of the small intestine some proteases that are basically stuck to the walls of our epithelial cells. So we refer to these as our brush border proteases. So here we can see enterokinase in the wall of our small intestine. And so the role of enterokinase here is it's going to be basically allowing us to have this active protease, trypsin. So trypsin does initially get secreted in this form trypsinogen. So we could think of that as like a zymogen, right? Trypsinogen would then get activated by enterokinase, an enzyme in the brush border, into trypsin. So remember that enzymes are proteins, which is why they are activated by proteases. So all the different proteases can get a little confusing here because we have enzymes that are breaking down the protein in our food, right? And we have enzymes like enterokinase that are helping us activate zymogens into enzymes that can help us break down our food. Okay. So another brush border protease that isn't on this diagram is Amino Peptidase. It has similar functions. Okay. So once we have broken down our amino acids using our different proteases, so specifically, let's just think about these pancreatic proteases, so trypsin, chymotrypsin, and carboxypeptidase, which are breaking the peptide bonds between our amino acids. Once we have our amino acids, so that just like individual monomers, they're going to be absorbed in the small intestine through secondary active transport linked with sodium mostly. Some of them will be able to enter the cells using facilitated diffusion. So there will be specific transporters, specific membrane proteins in the epithelial cells in the small intestine that are designed to allow amino acids through. They're then going to be able to cross into the plasma through facilitated diffusion, so they're going to move down a gradient. The absorption of dipeptides and tripeptides, so 2 or 3 amino acids linked together respectively, they're actually also able to cross into the cell, cross into the cells. So unlike our carbohydrates, which have to be in the form of monosaccharides, our proteins can be absorbed in slightly larger forms. So some dipeptides and tripeptides get absorbed as 2 or 3 monomers. And then they get broken down inside the cells to amino acids, ending up eventually with amino acids crossing our basolateral membrane into the plasma by facilitated diffusion, so down their gradients. Okay, so ultimately you're just going to end up with amino acid monomers in the plasma, but when they're crossing into the epithelial cells, they may be as 1 monomer or they may be 2 or 3 linked together. Okay, we will break here and we will talk about the breakdown of fats on Monday.